Immunotherapy has been already very popular in the hospitals to treat our cancer patients. Sadly, there's only a small fraction of these cancer patients respond very well to the current cancer immunotherapy. And we want to come up with new methods that can make these patients to respond again to the immunotherapies. I'm Li Tang. I'm an associate professor of bioengineering at the EPFL Lausanne. In my lab, we work on engineering our immune systems to better fight diseases such as cancer. This is a highly collaborative project, involves 3 labs. My lab will identify a compound that can stiffen the cancer cells specifically. Next, we'll send this compound to Camilla's lab, and she will test the killing by the human T cells of these treated cancer cells. And finally, in Oliver's lab, we are going to evaluate whether these physical features of cancer cells actually can be a biomarker that indicates how responsive will be for a specific patient. So the basic principle of immunotherapy is to take advantage of our immune system to fight cancer cells, rather than other therapies that are directly kill cancer cells, such as chemotherapies or radiotherapies. But there's a large area of research that's less appreciated, which is the physical properties of these cancer cells.
L'idée du professeur Li Tang a été de considérer la cellule cancéreuse non seulement sous l'aspect biologique ou chimique, mais plutôt sous son aspect mécanique et tout particulièrement sa plasticité cellulaire qui détermine combien une cellule cancéreuse est sujette à se faire attaquer par le système immunitaire.
We are going to using the imaging method to looking at the morphology of the cancer cells treated by various compounds. Very interestingly, we recently found that many of the cancer cells are actually softer than our normal cells. And even more interestingly, we found being soft actually make these cancer cells evade the surveillance of our immune system.
Now, when a T cell has to attack a cancer cell, it is better if it's stiff because the interaction is stronger. So by stiffening the cancer cells with our strategy, we aim at improving the interaction between a tumor cell and the T cells, and in this way improving the elimination of the cancer cells in patients.
De son côté, le professeur Olivier Miquelin va comparer les résultats des laboratoires de Camilla Jandus et de Li Tang avec des tissus prélevés sur de vrais patients.
The quality of the image is really fantastic. And do you see direct interaction between the T cells and the tumor cells in this sample?
At least at the edge, yes.
We are looking at proteins on the 2D tissue slides with a very high resolution, and this allows us to understand the biology of that sample and the way the immune system is interacting with the tumor. We will do this analytic here, but of course share the data with Camilla Jandus and Li Tang to be able to exploit the results together and to see whether what we are seeing in the patient is in line with what the models from Camilla and the discoveries from Li Tang are predicting.
So imagine if these cancer cells are very soft and the T cells come and are very difficult to grab and punch a hole, and this means that they are difficult to be killed by the T cells. On the opposite, if we have stiff cancer cells which have a higher surface tension, so when the T cells come and recognize these cancer cells, and it will be easier for the T cells to grab it and then eventually punch a hole and kill these cancer cells like this.
This cancer cell has been stiffened by our strategy, and so in this manner a T cell is able now to approach the cancer cells and to attach and also attack it and inject some molecules that at the end will lead to the destruction of the cancer cell, as you can see here.
The physical properties, in addition to the chemical, are indeed very important in the biological process, and there's a lot more to be discovered and also utilized for the therapies in the clinic.